Hey, how's it going, everyone? This is FixerMed. Welcome to my high-yield immunology review series for the USMLE Step 1, NBME CBSE, and NBME CAS examinations. This will be part 10 of a multi-part video series covering the entire discipline of immunology in a high-yield but comprehensive manner. So without further ado, let's go ahead and get started on today's content. So first off, I'd like to open up talking about the complement cascade. Complement proteins are integral to the innate immune system and are acute phase proteins synthesized by the liver. Three main pathways activate complement: the classic pathway, the alternative pathway, and the lectin pathway. These pathways lead to target cell death through the formation of the membrane attack complex, known as the MAC complex as well, or via opsonization and phagocytosis. Let's go ahead and move on to the next set of content. Key pathways and activation mechanisms. Classic pathway is initiated by complement C1 binding to the Fc region of IgM or IgG, leading to opsonization or cell death. Alternative pathway is triggered by C3b binding directly to microbial cell surfaces. Lectin pathway is initiated by mannose binding lectin binding to microbial surfaces. Moving on. Clinical significance and implications of this. So, a reduction in C3 levels is observed in conditions like post-streptococcal glomerulonephritis, known as PSGN, and systemic lupus flares. C4 levels decrease in cryoglobulinemia, further discussed later. C3A, C4A, and C5A are termed anaphylatoxins, capable of inducing mast cell degranulation and triggering anaphylaxis. C5A acts as a neutrophilic chemotactic molecule alongside IL-8, LTB4, kallikrein, platelet activating factor, and bacterial products. Deficiency in terminal complement components, C5 to C9, is associated with recurrent Neisseria infections, including both gonococcal and meningococcal. Hereditary angioedema is linked to C1 esterase inhibitor deficiency, not to be confused with C1 esterase itself. Paroxysmal nocturnal hemoglobinuria results from increased complement-mediated lysis of red blood cells due to a deficiency in GP1 anchor, which anchors CD55, the decay acceleratory factor, and protectinin, CD59, to the RBC cell surface, providing protection against complement-mediated lysis. Alright, now that we have a good introduction and baseline, let's go ahead and take a look at some questions. A 34-year-old patient presents with recurrent Neisseria infections, including both gonococcal and meningococcal infections. Laboratory investigations reveal a deficiency in terminal complement components, C5 to C9. Which of the following statements regarding complement activation is most accurate? Alright, I think I gave you guys enough time here. If you need more time, please feel free to pause the video. Otherwise, moving on now. Deficiency in terminal complement components, C5 to C9, results in impaired formation of the membrane attack complex, also known as the MAC complex, leading to reduced ability to lyse Neisseria bacteria. This deficiency is associated with an increased risk of recurrent Neisseria infections, including both gonococcal and meningococcal infections, due to the crucial role of the MAC in destroying these bacteria. Therefore, the correct answer is D. Deficiency in terminal complement components C5 to C9 is associated with reduced risk of recurrent Neisseria infection. Let's go ahead and take a look at why the other answer choices are incorrect. A. Complement proteins are primarily synthesized by the liver as acute phase proteins, not by neutrophils, which primarily play a role in phagocytosis and inflammation. B. The lectin pathway is initiated by mannose binding lectin, not by direct binding of C1 to the Fc region of IgM or IgG as described in the classic pathway. The classic pathway is initiated by complement C1 binding to the Fc region of IgM or IgG, not by mannose binding lectin binding to microbial surfaces, which is characteristic of the lectin pathway. E. While activation of the complement cascade can lead to target cell death through both the MAC and phagocytosis, the MAC primarily mediates target cell lysis, while phagocytosis is facilitated by opsonins like C3b and IgG, not IgG alone. Moving on to the next question. Patient with recurrent bacterial infections present with a deficiency in a pathway crucial for opsonization and cell death in the innate immune system. Which of the following is the most likely pathway affected in this patient? Alright, I think I gave you guys enough time here. If you need more time, please feel free to pause the video. Otherwise, moving on now. The alternative pathway of complement activation is triggered by C3v binding directly to microbial cell surfaces, leading to opsonization and cell death. Deficiency in this pathway would impair the patient's ability to efficiently opsonize pathogens and eliminate them, resulting in recurrent bacterial infections. Therefore, the correct answer is B, alternative pathway. Let's take a look at why the other answer choices are incorrect. The classic pathway is initiated by complement C1 binding to the Fc region of IgM or IgG, leading to opsonization or cell death. Deficiency in this pathway would impair opsonization and cell death, but is less likely to present with recurrent bacterial infections compared to deficiency in the alternative pathway. The lectin pathway is initiated by mannose binding lectin binding to microbial surfaces. While deficiency in this pathway could impair opsonization and cell death, it is less common than deficiency in the alternative pathway and is not typically associated with recurrent bacterial infections. Toll-like receptor pathway is not directly involved in complement activation, but rather plays a role in the recognition of microbial pathogens by the innate immune system. The phagocytic pathway involves the process of phagocytosis by immune cells, which is downstream of complement activation. Deficiency in this pathway would not directly impair complement activation and is less likely to present with recurrent bacterial infections. Let's go ahead and take a look at the next question. A patient presents with recurrent bacterial infections and episodes of angioedema. Laboratory investigations reveal reduced levels of certain complement components. Which of the following complement deficiency is most likely responsible for the observed clinical manifestations? Alright, I think I gave you guys enough time here. If you need more time, please feel free to pause the video. Otherwise, moving on now. Deficiency in C1 esterase inhibitor is associated with hereditary angioedema characterized by recurrent episodes of angioedema. This deficiency leads to uncontrolled activation of the complement system, particularly the kinin pathway, resulting in excessive production of bradykinin, which causes increased vascular permeability and angioedema. Therefore, the correct answer is C, deficiency in C1 esterase inhibitor. Let's go ahead and take a look at why the other answer choices are incorrect. Reduction in C3 levels is observed in conditions like post-streptococcal glomerulonephritis, PSGN, and SLE flares, systemic lupus flares, erythematous, but is not typically associated with recurrent bacterial infections or angioedema. Reduction in C4 levels is observed in cryoglobulinemia, but is not typically associated with recurrent bacterial infections or angioedema. Deficiency in terminal complement components, C5 to C9 is associated with recurrent Neisseria infections, including both gonococcal and meningococcal, but is not typically associated with angioedema. Deficiency in C3A, C4A, and C5A, termed anaphylotoxomes, can induce mast cell degranulation and trigger anaphylaxis, but is not specifically associated with recurrent bacterial infections or angioedema. That will do it all for the video today, guys. If you have any questions, comments, or concerns, please feel free to leave them in the comment section below. As always, would appreciate if you could like and subscribe to this channel for more content like this. But anyways, this is FixerMed signing off. Be sure to have a great day, everyone, and good luck studying. Goodbye.